So welcome learners in this session on synthetic seed technology. In this video tutorial, we will talk about what is synthetic seed technology, a technology which is considered to be an alternative approach for clonal propagation to avoid heterozygosity. We will also discuss how one can synthesize synthetic seed in laboratory condition and also its application. And on completion of this session, you will be able to analyze problems related to mass multiplication for genetically similar plants using synthetic seed technology. In some crop species, seeds propagation has been successfully, mainly due to heterozygosity of the seed. The seeds are very minute, the presence of reduced endosperm, requirement of seed with mycorrhizal fungal association for germination in case of orchids, and seedless variety of the crops like grapes, watermelon, and some other crops which produces seedless fruits, and the polyploidy label, which also affect the seed setting and propagation. So because of these problems, the seeds of many crops cannot be used for raising the further generation. And when the requirement is the mass multiplication of genetically similar plants, then seed propagation is not a preferred method. Now, what is the need of synthetic seed technology? Some of these species can be propagated by vegetative means. Species means those who has seeds, but seeds are not viable, or seeds leads to genetic variability. So maintain the genetic similarity. These species are propagated by vegetative means, but the vegetative propagation is slow and a cumbersome process. However, in vitro mass multiplications are time consuming and expensive too. And uniformity is the problem in both the cases, because in the presence of pre-existing genetic variability in the somatic cells, because of chromosomal aberration and other reasons, even the somatic cells leads to the genetic variability. And why? What is the need? What is the advantage of the synthetic seed when natural seeds are already available? So we have outlined some of the background issues in previous slides. Now the synthetic seed, it offers direct delivery of the tissue culture plant to the field conditions, which is not possible in any of the in vitro methods. It ensures the propagation of desirable genotype with genetic uniformity, which is very much required. Reduction in cost of the vegetative propagated elite plants. It also ensures the preservation of germplasm and convenience of germplasm exchange and transportation. It ensures the reduction in seed dormancy, because the in vitro methods, the environmental condition, cultural condition can be manipulated and controlled by automation. And it ensures the large production of identical embryos in short time, which is very much required, and it can produce throughout the year without any seasonal constraints and variations. Now, I have tried to compare the synthetic seed technology and its outcome with a conventional method of propagation. So if you see the conventional methods, you'll find that the conventional methods, it leads to the low volume production of the planting material. So these are considered as a small scale propagation methods. The maintenance of genetic variability is very, very difficult. The rooting of the plantlets required prior to the field condition. High cost per plant because sprout cannot be produced in large amount, and the multiplication rate limits by the mother X plants. So mother X plants is another limiting factor in case of conventional methods. When we compare this to the micropropagation, which is in vitro method, we found that the root induction requires prior to the field condition in micropropagation. The root induction followed by the acclimatization, which is a very, very critical and crucial phase through which the mortality, because of the high mortality, only 50% of the plantlets can be transferred to the field condition. And because of the mortality and involvement of various chemicals, glasswares, sophisticated culture conditions, the cost is very high per plant. And relatively, the multiplication rate is very low. Whereas in case of a synthetic seed, if you evaluate, you'll find that it offers a large scale propagation method. It maintains the genetic uniformity of the plant to largest extent. Direct delivery of produce to the field is the most advantageous part of it. No prior root formation is required because synthetic seed are produced by somatic embryos. And as we have already discussed, the somatic embryos are biplorar structure. So there is no requirement of root induction or shoot induction prior to the transfer to the field condition. There is no acclimatization process involved because germination is itself in the field conditions. Because of the large production, the cost is low and the rapid multiplication of the plant can be achieved. So these are the advantages of synthetic seed over micropropagation, that is in vitro methods and the conventional methods. Now, the concept of the synthetic seed was proposed by the Muraching in 1978. The first synthetic seed was produced by Keto and Jennings in 1982 using carrot somatic embryos. Here, in this picture, you can see a normal seed, natural seed, and synthetic seed. The synthetic seed is natural seed-like structure derived from the somatic embryos in vitro culture after encapsulation in the hydrogel. Such seeds are encapsulated in a protective gel, that is calcium alginate, against microbes and which protect it against microbes and desiccation. Now in this picture, you can see the synthetic seed and compare it with the natural seed. So in general, in a broad sense, if you see the structure anatomy of the natural seed, you'll find that there is a seed coat which gives protection to the seed from environmental stresses, microbes, and desiccation. There is an embryo which was formed after the fertilization of gametes from parents. And there is a food source which is known as endosperm. When you compare it, you will find that in synthetic seed, there is an artificial seed coat, which is constructed in the lab. There is artificial endosperm, which is sodium alginate, and there is an embryo, but this embryo is not produced by normal sexual cycle, sexual reproduction. It is a somatic embryo, which was derived from the somatic cells using in vitro culture techniques. So both the structure is exactly same, except for the natural seeds are produced in nature through sexual reproduction, whereas the somatic seeds, the somatic embryos are taken, and rest of the things, artificial food source, that is artificial endosperm, and the seed coat is synthesized in laboratory condition. Just to recap, that we use somatic embryo for production of artificial seeds, as we have already discussed in previous tutorials. The somatic embryo is the embryo which is generated from by using the in vitro techniques. It can be generated from somatic cells, and the somatic embryo has different stages. This is the globular stage, that it matures to the heart-shaped stage, then torpedo stage, and finally cotyledonary stage. Here in this picture, you can see the somatic embryo, copybook style picture at the top, and these two pictures down below. These are my experiments with the medicinal plants. I have raised the somatic embryos, and you can see the actual real pictures, and you can compare it. So in the synthetic seed, you can see in this picture, just to give you an idea what is synthetic seed, there is a matrix that is composed of organic and inorganic nutrients, some antibiotics, plant growth regulators, and its substitute of endosperm of natural seed. Then there is a propagule. It is bipolar somatic embryo structure, unipolar shoot tip, nodes, and that is substitute of zygotic embryo. And there is a hard covering outside, made up of calcium alginate produced by ion exchange reactions and on sodium alginate and calcium nitrate and chlorides. That is substitute of seed coat. So this is the simple structure of synthetic seed. Now you must have got some idea what is synthetic seed and how it can be developed and how it is a substitute of natural seed. Now, why is alginate capsulation the most suitable matrix? So the sodium alginate is frequently selected as a matrix for synthetic seed because of its moderate viscosity, low spinability of the solution, low toxicity to the embryos because you are dealing with a living structure, quick gelling agent, cost is very low, and biocompatibility with characteristics. That is why this sodium alginate or alginate is being used as encapsulation medium for most cases of production of synthetic seed. So the somatic embryos, as you know, lack seed coat and endosperm that provides production and nutrition for zygotic embryo and developed seed, as we have seen in the last picture. So to augment this deficiency, addition of the nutrition source is desired to encapsulation matrix. So nutrition source, salt, carbon source, and the growth regulators that are mixed in the gelling agent and that serve as artificial endosperm to the synthetic seed. In addition to that, to prevent the embryo from desiccation and mechanical injury, a number of useful materials may be incorporated into the capsulation matrix. And these materials are the fungicides to protect from fungal infection, and there are pesticides, there are antibiotics to protect the growth of bacterial spores, and some mycorrhizal associations are also incorporated, which helps in providing the nutrition and also give symbiotic relationship to the organ. In addition to that, the activated charcoal is added to improve the convergence and vigor of the encapsulated somatic embryo. The charcoal breaks up the alginate and thus increase the respiration to the somatic embryo. Charcoal retains nutrition within the hydrogel capsule and slowly release them to the growing embryo. So it is also added as an adjuvant in the matrix. Then there is a complexing agent. In complexing agents, normally calcium chloride or potassium chloride or ammonium chloride is being used, whereas calcium chloride is used as a complexing agent in most of the cases. So once mixture of the sodium alginate plus somatic embryo plus adjuvant plus activated charcoal is added in drops in calcium chloride solution, beads are formed due to the ion exchange between the sodium ion, sodium alginate, and calcium ion and calcium chloride. The hardness of the synthetic seed that is formed is independent on number of calcium ion and sodium ion. So this calcium chloride, which provides the hardness of outer covering of the synthetic seed, you may compare it as a seed coat of the natural seed. So this is a substitute of the seed coat, which provides protection as well as protect the synthetic seed from environmental stresses. So for optimum hardness is very much required. of the wheat, the concentration of sodium alginate and the calcium, and the time required for the complexing should be optimized in the experimentation. So in case of various production of various types of synthetic seed, this has to be optimized in the culture condition. Normally, the sodium alginate 3% and calcium chloride 1% are used, and the compaction time is half an hour is reported in various literature. So by maintaining these conditions, the optimum hardness can be achieved. Now here in this flowgram diagram, you can see the production of synthetic seed. First, the callus culture is raised by taking any of the explant. The next plant is incubated in semi-solid culture media, Morashing and spruce culture media, fortified with cytokinin and auxin in equal ratio. Then callus is achieved, which is an unorganized mass. Then after putting the callus on 2,4-D for a longer period of time, and when it is 2,4-D is removed from the culture, somatic embryos are start producing, and these somatic embryos are started generating from a single cell of the uppermost layer of the cell of the callus. Some of the cells become embryogenic and they start producing embryo-like structure. This embryo-like structure, as we have already discussed, these are also called embryoids, which is a bipolar structure. One pole, produce shoots, and another pole produce shoots. Now this can be excised from the callus and mixed in sodium alginate solution. So as we have already discussed, this sodium alginate solution, here in this picture you can see sodium alginate solution. This contains insecticides, some pesticides, some micro antibiotics, and source of energy, and also the activated charcoal. When we mix it, and it serves as the source of food source as a substitute in case of endosperm in natural seeds. And after that, these beads are put into the calcium chloride solution in drops. The calcium ion reacts with the sodium ion and makes a hard covering around each seed. And after sometimes after gelling, the seeds covered under a synthetic seed coat, and these seeds can be put onto culture media, which is having no hormones because of the bipolar nature of the somatic embryos. The somatic embryos germinates and produce a food plant, and this plant can be transferred to the field condition. Even there is no need to follow this step. This can also directly transfer to the field condition in a sterile soil first, and then can be transferred to the field condition, and that can be germinate into the full plant. So you can see the seeds a synthetic seed can be used exactly the same way the natural seed can be produced. So the procedure I have chalked out once again, just to give you some more in-depth idea. So the first establishment of somatic embryo, followed by maturation of the somatic embryo, and synchronize and simulate the somatic embryo, because synchronization is very, very important, and synchronization has been achieved. That we have already discussed the technique in previous tutorials. When the mass production of somatic embryo has to be achieved, then standardization of the encapsulation process and standardization of artificial endosperm and mass production of synthetic seed can be achieved. And that seed can be grown in glass house or in field condition onto natural soil. There's no need to germinate it on culture media. Here, the encapsulation process is, described through a culture through a flow chart, which is exactly same as we have discussed in the previous slides. Some automation of the encapsulation process has also been done. So this picture is not very much clear, but by going through the literature, you can get a better representative picture. In this picture, this is the quick method of artificial seed production based on the basic technology that we have already discussed. In this picture, you can see the alginate solutions with embryo is fed from a supply tank. The alginate capsules were planted in feeding tray using a vacuum seeder. The capsules are planted in the field using a stand head plate, and a hydraulic coating is required for the mechanical handling. So by using this automated system, the synthetic seed can be produced and large mass of synthetic seed can be achieved. Now, so far we have understand the basic techniques of the synthetic seed. Now, there are two types of synthetic seeds based on its production methods. The synthetic seed depends on the nature of the planting material, can also be of two types. First is the hydrated seed, synthetic seed, or second type is desiccated synthetic seed. So hydrated synthetic seed consists of somatic embryos which are encapsulated in a gel which remain hydrated throughout. Such synthetic seeds are produced in plant where the somatic embryos cannot withstand the desiccation. So it is genotype dependent and the species dependent also. Whereas the desiccated synthetic seeds consist of somatic embryo, which are hardened, mature to withstand desiccation, and then capsulated in a suitable coating gel. The viability of this synthetic seed is tested at a regular basis. So these two types of synthetic seeds are being produced, and this is because of the genotypic difference of various planting materials, and some of the seed they can withstand desiccation, and some of the seed that cannot tolerate the desiccation, then hydrated methods can be used. Here once again you can see the one synthetic seed which is produced by using the same technology that we have already discussed. Here in this picture you can see the synthetic seeds which is encapsulated in sodium alginate and germinating synthetic seed picture. So the artificial seed production and the plant conservation. In this slide, you can see that somatic synthetic seeds have been produced by somatic embryos in case of papaya, brinjal, mango, carrot, sandalwood, alfalfa, lettuce, and celery. These are the success stories. In these crops, the synthetic seeds were produced using the somatic embryo. But in addition to the somatic embryos, the auxiliary bud and the shoot tip has also been used in some cases. In case of the eucalyptus, the citrus, and the pineapple, there are reports where the auxiliary bud or adventitious bud, which is dormant at the axil, that has also been used as a propagule for production of the synthetic seed. Even in some cases like banana and cardamom and apple and the kiwi, the shoot tip is used as a propagule for production of the synthetic seed. So a lot of meristematic parts can be used for production of the synthetic seed. But by and large, somatic embryos are the most amenable propagule for the production of the artificial seeds. Here in this picture, you can see the production of a synthetic seed in papaya. This picture is also original. This is one of my PhD students who has worked on papaya somatic embryogenesis. And These embryos can be used for synthetic seed production. Various developmental stages during plant regeneration via artificial trees in Caraca papaya, production of somatic embryo, then encapsulation, then incubation in the culture media, and generation of the plantlets from the synthetic seed, and these plantlets can be transferred to the field condition after placing it in sometimes in polyhouse and glasshouse. In this picture, you can see the somatic embryos of the banana for encapsulation. And in this case, the somatic embryos were, encapsulated in 5% sodium alginate. Then the citrus, the technology is very, very successful in case of citrus. And what are the basic advantages, major advantages of the synthetic seed over the natural seeds? So the artificial seeds or the synthetic seeds are easy to handle and easy to transport, and transport charges are very low due to the small size and due to the covering of a small space. The artificial seeds has potential for long-term storage without losing viability. So that is why these are the important source of germplasm conservation for superior or extinct and endangered species. The synthetic seed production is cost-effective when compared to the other methods, as we have already discussed in initial slides. It allows the economical mass propagation of elite plant varieties, because for mass production of the elite plant varieties, genetic similarity is very, very important. So these varieties cannot be further multiplicated through natural seeds because of the intermixing of the characters. And in some cases, that asexual reproduction or the conventional method has its own constraints. So for multiplication, this method, the synthetic seed production and multiplication, is the most suitable method for such purposes. The synthetic seeds are of an immense importance in species which do not produce viable seed. Very well understood. The propagation of hybrid plants is very easy through artificial seeds. So genetically modified crops and endangered species of the plants can be propagated through artificial seed technology. Here, I would like to further elaborate this thing, genetically modified crops, because the micropropagation, the somatic embryogenesis can also be used as an source plant material for genetic modification or the transformation experiments. In the case of papaya, we have experimented it. We have used the somatic embryo for transformation through Agrobacterium tumefission, and we have achieved the significant number of transformation by using the somatic embryo. So any gene of the interest can be transferred to the somatic embryo, thereby the genetic manipulation can be done, and those characters will be further expressed when the synthetic seed produced by this transformed somatic embryo when transferred to the field condition. It is very, very successful in germplasm conservation also, because it provides the long-term storage. So these are the basic, very important artificial application of the synthetic seeds. It also, ensures the genetic uniformity of the crop, and varieties of the crops can be easily maintained. These artificial seeds provide disease-free condition to the planting material, because as an adjuvant, we mix antibiotics, antifungal, and other chemicals which makes them disease-free, completely disease-free. So during the production of artificial seeds, we also mix herbicides that can be added to the formulation. These herbicides to provide extra protection to the plants against the pests and diseases. And the synthetic seeds crops are easy to maintain because of uniformity in genetic constituents. So these are the important application of genetically artificial seeds. Now, for the further reading, you can consult a book, An Introduction to the Plant Tissue Culture, edited by M.K. Razdan. You can also discuss the topic, read the topic in a book named Plant Tissue Culture, Theory and Practices, edited by Bhojwani and Razdan. And I will also recommend one review article, which is written on the synthetic seed. A DOI number has been mentioned here. In addition to this, you can consult the ePathshala. The chapter name is In Situ Conservation and Cryopreservation, where there is a very good detail on synthetic seed production technology. You can just log on to this ePathshala using this web link. The registration is free of cost, and this ICT initiative, Ministry of MHRD, is offers more than 22,000 lectures and the tutorials and the study materials for UG and PG courses. You must explore this thing. Apart from all above, I have already uploaded the e-content on LMS and also uploaded the e-content and the material on MS Teams, File Menu, and on MS Streams also. And after completion of this tutorial, I will also upload this video recording on LMS, which you can go through and revise the topic. With this thing, I think you are now able to synthesize your knowledge to solve some of the issues which are related to mass multiplication of genetic similar plantlets by using the synthetic seed technology. With this, I stop here and wish you all the best. You can go through the e-content that I have already shared for further reading. Thank you very much for your patience.